Hi students, this is Sayan. Welcome to my biology class. My channel will help you to build thorough knowledge on biological topics and also make you avail all important notes for exam purposes. So let's get started. Today we will discuss about polymerase chain reaction, simply known as PCR. So at first, let's know about its definition. It is a molecular technique used as an alternative to gene cloning. With this technique, any particular segment of DNA can be amplified, make millions of copies. This technique is nowadays useful technique for medical and biological research labs. So, what is the basic of this concept? If you take any short segment of DNA, you will able to produce millions of copies of this particular DNA. So, PCR will make you able to construct several million of copies of a single segment of DNA. Now, let us know about its history. This technique PCR was discovered for the first time by an American scientist, Carey Mullis, in the year of 1983, and he was awarded with a Nobel Prize in the year of 1993. But we should all know that this concept of PCR was already been discovered by Hargobind Khorana and his colleague Kleppe in the year of 1966. Although their name was quite different, they put the name as repair replication. So, it is the Hargobind Khorana and Kleppe who introduced the concept of polymerase chain reaction like process for the first time, and later on Carey Mullis modified this technique and put forward the application of PCR in front of the whole world. Now, let us move to the next topic. Ingredients required to set up PCR. We should all know that what type of ingredients we should take to start the process of PCR. Here are those ingredients. First of all, we have to take DNA segments to be amplified. The short segment of DNA which are to be amplified by the process. Then we need to add primer. What are primer? Primer are artificially designed oligonucleotides. Those can attach to any target DNA. So, primer are very important things for this technique of PCR. Then we have to take DNA nucleotide bases, simply known as DNTPs. And for this purpose, we have to take adenine, guanine, cytosine, thymine bases. Now next, we have to take buffer. Buffer are required to ensure right condition for the reaction. And last but not the least, we have to take a special polymerase known as Tec Polymerase. It is a thermostable enzyme. It can polymerize the DNA at temperature higher about 70 to 80 degrees Celsius. So, it is highly heat stable enzyme and it can perform its entire activity even at this very high temperature. For the first time, this enzyme Tec polymerase was isolated from the bacteria Thermus aquaticus. It is a thermophilic bacteria. This is the only enzyme in the living world that can perform at this very high temperature. Otherwise, we all know that human DNA polymerase can act at around 37 degrees Celsius. So, in that respect, Tec polymerase is highly useful for the PCR. So, next we move to the next topic. Let us find the principle of the technique. What are that? This technique is based on the principle of thermal cycling. What is thermal cycling? A process of periodic heating and cooling carried out by machine. So, it is an entire machine based technique. All you have to design and program the machine prior to start the technique, and then the machine will automatically carry out the thermal cycling within a fixed period of time, and thus it can automatically produce multiple copies of DNA. Next, we move to the next topic, steps. What are the steps of PCR? If we conduct an experiment with PCR, we should know that these are three basic steps. First, number one, denaturation. Denaturation of DNA. That means breakdown of double stranded DNA into single stranded DNA. Then annealing. Annealing or attachment of primer. Attachment of the primers with the single stranded DNA. And next, extension or elongation of primers by Tec polymerase. So, these are the three steps. Next, we should move about the procedure of the technique. Here it is a diagrammatic representation which help you to understand the entire procedure. Let us start about the procedure. Suppose this is the original DNA we have taken. This is a double stranded DNA. So, at the first step we have to break down this double stranded DNA into single stranded DNA. So, we have to undergo for denaturation. Denaturation is a process which will split the double stranded DNA into single stranded DNA, and we have to attain about 94 to 95 degrees centigrade of temperature for that purpose. We have to keep the entire solution for 15 to 30 seconds. So, after that the double stranded DNA will easily get breakdown into single stranded DNA. Then what after that? After denaturation we have to follow the next step, annealing. Annealing of primer. Here in this diagram we can easily find that these primers are being attached with the DNA. For attachment of primers we have to attain a temperature around 50 to 60 degrees Celsius, and always remember the primer will get attached at the 3' end of the existing single stranded DNA, and holding the 3' hydroxyl end, Tec polymerase will extend the primer towards this direction. We have to keep the entire solution for next 10 to 30 seconds. After that, extension of primer takes place by the Tec polymerase. Here in this diagram, we can easily find that the primer has been extended by the Tec polymerase, and for this purpose we have to keep the solution at 72 degrees Celsius for 2 minutes. Thus, a single cycle of PCR has been completed, and I hope that students will find this entire procedure diagram very useful for them. Now, we will move to our next topic. What are the applications of PCR? Why should we perform PCR? PCR helps us in many ways. At first, it helps to detect pathogens even in very small quantity in our blood. Next, it helps prenatal diagnosis for any genetic disease in human beings. It also helps in DNA fingerprinting. Suppose if you are working with a very little amount of DNA, then you can easily amplify the DNA content in many millions of time, and then you can perform your own DNA fingerprinting procedure. This technique of PCR is also helpful for gene therapy and other kind of biological researches. This technique of PCR is also helpful in paleontological survey, in forensic science, and especially in cell-free molecular cloning. So these are few applications of PCR technique. Thus, with this entire video, we can perform or we can know the entire basic concept of PCR. I hope students will make this video very useful for them, and I hope you will easily love this video. If you really so please don't forget to like this video and subscribe my channel. Thank you and bye.